Hello, I'm Peter Langridge and I've been very actively involved in the Barley Genome sequencing project. The Barley sequencing project was set up quite a few years ago, and the objective of it was to try and establish a reference sequence for the entire Barley genome. And this is quite a challenging task because the Barley genome is about twice the size of the human genome. So because the Barley genome is so very large, as I said, twice the size of the human genome, we need to really come up with a very special strategy or approach to try and do the sequencing, or to assemble a useful sequence that we could then work with. And the reason why we needed a sequence to work with, because we need to understand where the genes are positioned, how the genes interacted with one another, and how they behaved both in a normal barley plant, but how they behave during a breeding program when breeders are trying to mix genes from different sources together. So a particular issue we had in trying to sequence the barley genome was to really understand where the genes are positioned and how they interacted with one another. There are between 30 and 40,000 genes in the entire barley genome, and those are spread out over the 7 chromosomes of barley. That means each chromosome has in the order of 10 to 15,000 genes. So the position of the genes and the way they behave becomes really important when breeders are making crosses and trying to select for new progeny that have an interesting new combination of genes from the two parents. However, because the genome is so very large, it consists not only of the sequences that encode the genes themselves, which then ultimately affect the phenotype and characteristics of the plants, but also a whole range of other sequences that are generally referred to as junk DNA, although it's not really junk. The way in which the genes behave is very important relative to all of this extra information, this junk DNA. And trying to work out that relationship becomes very important because it affects the way in which genes recombine or mix in the progeny. So for example, if a gene is located out towards the tip of a chromosome, it will recombine much more readily than a gene that's located close to the centromere, which is around the center of the chromosome, and a region where there's very low recombination rates. That information is absolutely crucial to a breeder to make a decision about what possible combinations are likely to come out of particular crosses. And it also tells us whether we need to focus on particular regions of the genome, of the chromosome, in order to try and increase recombination. So can we get a good event occurring if we have 100 progeny, or do we need 10,000 progeny in order to have a proper recombination event? So another really important aspect of having this essentially gene map of barley, and this is both a genetic and a physical map, should explain that a genetic map is based upon the recombination rates between genes, and the physical map is related to actual physical position along a chromosome. And these two are not the same, and that's where the whole issue of recombination comes into play. But the other important aspect of all of this is that we can use genetic information to try and zero in on a physical position on a chromosome. And the physical position ultimately will give us the gene. If we have the gene, then we can actually start asking questions about how the gene behaves, how it functions in a normal plant, and we can also start looking for novel variants of the gene. For example, we can go to some of the wild relatives of barley, or some of the land races that are still available in germplasm collections, and see if we can find novel variants that may actually have improved characteristics if they're introduced into our elite lines. So I mentioned that having this sequence or this structure or arrangement of the genes helps us isolate genes and lets us find novel variants. This is really important because plant breeding is based upon the utilisation of variation. But breeders tend to do most of their work within elite germplasm when they've got a high probability of getting a really good outcome from a particular cross. What we can do if we have a knowledge of the genes is allow them to do this in a very targeted fashion and say, here is a wild relative of barley, there's a particular region of the genome that could be really useful in your breeding program. What you now need to do is to target that region and introgress that. That means recombine that into elite germplasm to capture those benefits. So it allows us to really expand the variations available to breeders. And that ultimately translates to benefits to growers. Because if we can get new variation in there, new sources of disease resistance, new sources of enhanced quality, we can try and achieve those step changes in breeding that have been so crucial to the success of the industry in Australia. There's a third important aspect of this work as well, and that is the relationship between barley and wheat. These are two very closely related species, and a lot of what we learn in barley we can then translate across into wheat. So for example, a gene we discover in barley is probably going to be present in a similar position in the wheat genome. But wheat is 3 times bigger than barley, so the problem we have with barley being twice the size of the human genome is amplified still further when we go to wheat. So barley provides a really good model for trying to understand genes, gene behaviour, and then translating that information across into wheat. So we're really in a great position now. We've got a physical map, we've got a really good genetic map of barley. We know where pretty well all of the genes sit, both physically and genetically, and we can use that to try and understand both the way the genes behave and the structure of the genome. But it's not a complete sequence of the barley genome. So that's our next task, is actually to assemble the sequence, the exact sequence all along all of these chromosomes. And we're now well down the path to be able to do that. This map that we've now created is going to form the base of the sequencing. And under current plans, it's hoped that this international consortium that's been working on this will have the full sequence completed by the end of next year.